Hi everyone, my name is Gwendolyn Pyeatt and I work in Dr. Karen Allen's lab at Boston University. Today I'll be presenting my work on the structural characterization of the covalent binding of Keap1 and thiol-containing small molecules. So the overall goal of my project was to design a protein-protein interaction or PPI inhibitor that combats oxidative stress by targeting the interaction between proteins Keap1 and Nrf2. So first, as some background, oxidative stress is characterized by high levels of reactive oxygen species, or ROS, which include things such as hydrogen peroxide, superoxide, and hydroxyl radical. Sources of ROS can either be external from things such as carcinogens and radiation, or internal from processes like inflammation and cell signaling. These all cause ROS levels in the body to increase, thus triggering oxidative stress. Since these reactive oxygen species can be damaging to proteins, lipids, and DNA, oxidative stress is associated with many diseases, such as diabetes, cancer, and neurodegenerative diseases like Alzheimer's and Parkinson's disease. Kelch-like ECH-associated protein 1, or Keap1, is a major regulator of the body's response to oxidative stress. Under conditions without oxidative stress, Keap1 forms a homodimer via its bric-a-brac, tram-track, and broad complex, or BTB domain, and then with its two Kelch domains binds two separate motifs of transcription factor, nuclear factor erythroid 2-related factor 2, or Nrf2. Shown here is the structure of the Keap1 Kelch domain, which I expressed and purified in all of the studies I'll mention in this presentation. So this Keap1 homodimer under conditions without oxidative stress also binds Col3, an E3 ubiquitin ligase that targets Nrf2 for ubiquitination and proteasomal degradation. However, under oxidative stress conditions, sulfhydryl residues in the BTB domain of Keap1 are modified, causing it to release Nrf2, which can then translocate to the nucleus, bind to MAF protein, and upregulate the transcription of antioxidant enzymes by binding the antioxidant response element, or ARE. These antioxidant enzymes provide an efficient catalytic detoxification of reactive oxygen species, thus making the Keap1-Nrf2 interaction a good drug target for oxidative stress-related diseases. However, protein-protein interactions often take place between large flat interfaces on the two participating proteins, making them difficult to block. One method that can be used to target PPIs is fragment-based drug discovery. As opposed to traditional high-throughput screening, fragment-based drug discovery utilizes fragments, typically less than 300 Dalton in molecular weight, to probe the protein interface for what are called drug-hole binding sites, or pockets on the protein surface that can bind these fragments with high ligand efficiency. These fragments, while they may bind weaker than the small molecule drugs used in traditional high-throughput screening, often exhibit high binding efficiency, or binding energy divided by molecular weight. These fragments can then be elaborated into larger molecules to increase their potency and specificity in a process termed scaffold optimization. The method I used in my studies is called covalent disulfide trapping, a site-directed form of fragment-based drug discovery that utilizes a native or engineered cysteine residue on the target protein. How this works is what is known as the fragment warhead of the molecule will interact with first the protein surface, thus bringing it in close enough proximity to the tethering cysteine for a disulfide exchange to occur, thus tethering the fragment into place. This is especially useful for studying these complexes by X-ray crystallography, since it can be difficult to obtain structures of the protein complexes due to the weak binding of these fragments. A previous study by our collaborators at UCSF in the Arkin lab identified 43 of these thiol-containing compounds, which we call monophores, that can be tethered to Keap1 at a cysteine residue near the Nrf2 binding site. So that cysteine residue is shown here, cysteine 434, and in pink here is the Nrf2 peptide. So this is very close to this binding site right here. These monophores are designed with three functional elements. First, the fragment warhead I discussed earlier, which interacts with the protein surface. Second, the linker fragment that contains the thiol for tethering. And finally, a solubilizing cap, which facilitates disulfide exchange as well as keeping the monophore soluble in aqueous solution. Since the fragment warhead of these 43 identified monophores has to form a favorable interaction with the protein Keap1 in order to be tethered, it is likely that these monophores contain chemical properties that could be incorporated into the design of an effective, non-covalent, small molecule Keap1 and Nrf2 inhibitor. So under this overall goal of designing a PPI inhibitor that combats oxidative stress by targeting the Keap1 and Nrf2 interaction, one of my goals was to characterize the interactions between Keap1 and these monophores using X-ray crystallography to get structures of the protein-monophoric complexes, or adducts. This structural data could then be used to help guide the design of a new non-covalent Keap1 and Nrf2 inhibitor. So my objectives under this goal were first to identify new stabilizing buffer conditions in which these Keap1 monophore adducts could be formed, to then identify adduct crystallization conditions, obtain the structure of the adduct using X-ray crystallography, and then finally determine whether the monophore is favorably positioned for scaffold optimization. It was important to focus on this first point because the buffer condition we were using initially to form adduct caused the adduct to aggregate, making crystallization efforts very difficult. To address this issue, I first conducted a differential scanning fluorimetry, or DSF, buffer screen, which I measured the melting temperature of the Keap1 Kelch domain, which I will refer to as wild-type Keap1, in 96 different buffer conditions. It was found that the condition 50 millimolar EPS, 100 millimolar NaCl, pH 8.0 was the most stabilizing condition, which raised the melting temperature we typically observe for wild-type Keap1 in the size exclusion chromatography buffer by about 7 degrees Celsius. I then decided to move forward with this buffer condition with half the salt concentration, since a lower salt concentration is ideal for crystallography, and lowering the salt concentration only decreased the melting temperature by about 1 degree. We then wanted to see if we could use the DTNB assay to detect adduct formation in this new buffer condition. This assay can be used to measure reactive cysteine residues in a protein sample, which would include the tethering cysteine that is used to tether the monophore to the protein. So how this reaction works is first you incubate your protein with the reagent DTNB, and if you have reactive cysteine residues in your protein, it will undergo a simple disulfide exchange, thus forming this mixed disulfide product, and also TNB, which has a high molar absorptivity at 412 nanometers. You can then make a standard curve using just cysteine, and use this standard curve to calculate the number of reactive cysteine residues in your protein. I first optimized this assay in a high-throughput 96-well format to minimize the amount of protein I had to use in this new buffer condition I identified to optimize Keap1 stability in the assay. I was able to obtain a standard curve with a higher squared value, as well as a absorbance spectra with the expected isosbestic point of this assay of 356 nanometers. I then used this optimized assay to measure about 3 reactive cysteine residues for wild-type Keap1. So here I conducted the assay at 3 different concentrations and obtained a value of about 3 reactive cysteines. This agreed with structural data, since when I used PyMol to estimate the solvent-accessible surface areas of all 8 of the cysteine residues on the Keap1 Kelch domain, I found that 3 of them had a significantly higher solvent-accessible surface area than the rest, which would account for the 3 measured by the DTNB assay, since this is a time-dependent assay in which the cysteine residues react from most to least solvent exposed. I also confirmed the accuracy of the modified DTNB assay with a positive control of Keap1 denatured with 1% weight per volume, SDS, and I got a value of about 9 to 10 reactive cysteine residues, which is close to the known value of 8 reactive cysteine residues, or total cysteine residues. This error could be accounted for by either the hydrolysis of the reagent DTNB, or, which is common at the pH this assay takes place, or error in the protein concentration measurement. So overall, this did confirm that accuracy of the modified DTNB assay. I then wanted to see if there was a time at which I could take an endpoint and observe a change or a difference of 1 less reactive cysteine residue in a Keap1 monophore adduct as opposed to the wild-type control. This loss of 1 reactive cysteine residue would be expected in the adduct since the most solvent-exposed cysteine, the tethering cysteine, cysteine 434, would be lost upon adduct formation. So I first conducted a kinetic DTNB assay from 0 to 15 minutes, since 15 minutes is the incubation time used to measure the total number of reactive cysteine residues, which I used in the previous assays I mentioned. And I found that 5 minutes was a good time at which this endpoint could be measured to try to see a difference of one less reactive cysteine residue in the adduct. I then formed the adduct Keap1 bound to monophore 917833 in the original low DTT buffer condition we were using, in which adduct formation has been confirmed, and conducted this assay with a wild-type control with a 5-minute endpoint absorbance taken. And I was able to observe a difference of one less reactive cysteine residue in the adduct sample, which suggested that the DTNB assay can be used to detect adduct formation. I then tried to form the same adduct, Keap1 917833, in this new buffer condition I identified. And I conducted the same assay and was able to observe the same decrease of one reactive cysteine residue between the wild type and the adduct, which suggests that this adduct could be formed in my new buffer condition. So I then wanted to move forward with crystallization efforts of the Keap1 917833 adduct formed in these new EPS buffer condition, and I set up a high-throughput index screen which yielded 2 initial crystallization hits, and one of them being a quasi-crystal, which is shown here. So to summarize my work, I first optimized the DTNB assay to screen buffer conditions in which the Keap1 monophore adducts can form, which greatly accelerated crystallization efforts of these adducts, according to this procedure outlined here. I identified a buffer condition that stabilizes wild-type Keap1 and potentially allows the adduct Keap1 917833 to form, and I also identified 2 hits in a high-throughput index screen for the Keap1 917833 adduct, which can be optimized into diffraction quality crystals for structural determination. Future directions on my project would include first conducting MS-MALDI to confirm adduct formation in this new buffer condition, and also validate the DTNB assay as a method for confirming adduct formation, to then optimize the Keap1 917833 crystals to determine the structure of this adduct by X-ray crystallography, and finally to use this method to identify buffer conditions in which the other Keap1 monophore adducts could be formed, to ideally get crystal structures for all 43 monophores. This structural data could then be used to guide the design of a non-covalent small molecule Keap1 and Nrf2 inhibitor for the use of treating oxidative stress-related diseases. And with that, I conclude my presentation. I'd just like to thank my advisors, Professors Karen Allen and Adrian Whitty, as well as my graduate student mentors, Samantha Muellers, the rest of the Allen Lab, and of course, the Arnold and Mabel Beckman Foundation for contributing funding to this project. And that's all I have. Thanks for listening.